everyone. Thank you, Dr. Sharma and Dr. Shetty, for setting this up. This is shaped up beautifully into the discussion. So what we are going to talk over the next few minutes is we're going to link the studies on genetics and how we are moving towards gene therapy to bring this treatment in India. So in genetics, we know that over the past 3 decades, there's been an explosion of genetic studies that's led to the discovery of the genes underlying a variety of different conditions. And this is what you see on the screen is a genes to disease map. And you can see how dense these maps are. Each of these are nodes, and you can see retinitis pigmentosa, which is a huge number of genes associated with it, and you can see them linked with other diseases. And that's where you see the genotype-phenotype overlaps that we often talk about and that Professor Holder was also mentioning. So now that genetics has powered this, the genomics revolution followed, and we can now test people for it. So when are we going to get to the point of treating all of these patients? And that's where we had thought that the genomics revolution would lead to treatment for everything in the next 1 or 2 decades. But as it turns out, it's not quite so easy to select genes for doing treatments. And that's what we're going to discuss a lot about today. So inherited retinal diseases. So inherited retinal diseases really is the poster child for gene therapy. And that was one of the first gene therapies to be approved, right? So RP65 gene therapy for LCA2. So given the successes in inherited retinal diseases, so at the clinic, the retinal dystrophy clinic, we see a variety of in IRDs, and these present here with unique pathologic features. And we go on to do clinical assessments, genetic counseling and testing. And in the research side, we study the genotype-phenotype correlations. But what is our approach for personalized medicine for all of these treatment, all of these patients? So the key is to select the right treatment for the right patient. So how do we do the patient selection? So already you've heard Professor Holder talk about how to do that sort of work through ERG, and how do we follow these patients up and look for a functional correction. So these are the aspects that I now invite Dr. Poornachandra to talk about the work that is currently happening in Netralaya.
So thank you, Dr. Arka. Can I have the slide control?
Yes. You have it.
Yes, thank you, Dr. Arka. It was a kind introduction. You set up the tone. So I remember Professor Rohit Shetty telling once, I mean, in future, most of the disease in the body will have either a genetic etiology or a viral or immune modulation. So we as clinical clinicians, our responsibility is to generate a lot of this database for these clinical trials. So we have a registry of retinal dystrophies with more than 2,800 families who have been completely worked up. So we start from Leber's congenital amaurosis, and we have juvenile retinoschisis, dominant based, recessive based, achromatopsia, we have keratoconus, we have choroideremia, we have albipunctatus, retinitis pigmentosa, which is the most common as we were discussing, and Stargardt's, which is the most common macular dystrophy in this region. So basically all these varieties are also seen in India. So if you take one example of Stargardt's disease, so this is a nice cartoon which explains, see, Stargardt's is usually most of the time caused by a mutation in a gene called ABCA4. If you look at this, if his ABCA4 gene is fully functional, you can have a normal appearance of fundus, normal electrophysiological features, and a normal microenvironment between the retinal pigment epithelium and the photoreceptors. If it's partially functional, we get what we call the typical textbook description of Stargardt's, like flex, beaten bronze appearance and all. If it is severely dysfunctional, it can also have a clinical manifestations of cone dystrophy, cone rod dystrophy, and a retinitis pigmentosa. As Professor Holder was mentioning, it's the same gene, can have a different clinical manifestation. So to give a clinical example, so this patient was referred to me as a suspected dystrophy, where fundus appeared normal, autofluorescence did show some atrophic changes, which was same was confirmed when it did an OCT, which showed foveal thinning and the outer retinal loss. So this patient had a compound heterozygous mutation in ABCA4. You know, like Stargardt's is a recessive disease. The mutation has to be homozygous. This patient had compound heterozygous. So this patient was born out of a consanguineous marriage, which is again very common in this part of the world. So surprisingly, the normal mother also had this ABCA4 mutation. She was homozygous for one of these heterozygous genes. So she said she's normal. So I thought I would like to see the mother. So when I saw her clinically, so the fundus, as expected, appeared absolutely normal, normal vision, normal color vision, everything. But when I did an autofluorescence, there was something going on in the perifoveal region, and there was also an EZ disruption in the perifoveal region. The photoreceptors are quite intact in the foveola. That's why our vision is quite normal. So it's always important to screen the family members, even if they report they are normal. So this is another interesting family. This patient was referred to me as a case of Stargardt's disease. So, but looking at the autofluorescence and OCT, didn't appear to me like it's Stargardt's. She was given a Stargardt's diagnosis because father had a phenotype of Stargardt's. So when we did a genetic testing, we got a novel gene called SERPINF1. I have not displayed it here; it's not published yet. So we did a fulfilled ERG for her. If you look at the dark adapted responses, there was some amplitude reduction in the 30 and 10 ERG, and also in light adapted single flash and the flicker responses. So this was the father's fundus photo and autofluorescence and OCT, which can very well fit into a Stargardt's phenotype, as I was telling, with atrophic macular changes and a resolving flex slightly away from the areas of degeneration. And the proband, the lady had two children who also had mutations. So I thought, let me screen the children who are normal as stated by her. So I saw the children's fundus as expected. It was normal, and OCT also looked normal. But now look at the autofluorescence. So everything is now there is some abnormally hyper autofluorescence in the macular region, telling that there is some change in microenvironment, the way the retinal pigment epithelial cells are talking to the photoreceptor. There is something wrong here. So though they are, asymptomatic. So we went ahead and did an electrophysiological test. The daughter had more or less a normal scotopic responses and an amplitude reduction, light adapted single flash response. So we tried doing a pattern ERG. The right eye looked okay, left eye was abnormal. The fundus, a multifocal ERG also showed reduced central responses with reduced light peak. So this was the sibling compared to the daughter. This patient had a slightly more rod cone dysfunction, and we could not do a pattern ERG in that patient. We were able to capture that patient was very uncooperative for examination. So somehow we managed to get this multifocal ERG. We did show reduced central responses, though recording looks a bit noisy. So here we have a 3-generation, the grandchildren who are normal like this. Autofluorescence is showing abnormality, a mother and a grandfather. So all of them have the same mutation. So the fate of these children is written here. So eventually they are going to pass through this and end up here, which is likely. So if you want to stop this, we need something called a gene therapy to arrest the disease at the earliest stage. So before going to that aspect of it, so we also have something called a polarization sensitive OCT, which is an ultra high resolution OCT, which can generate NFAS image, which is custom built. So basically some of who are listening to it for the first time. So in addition to the standard OCT, this OCT also considers the depolarized state of the tissue. If it's more the functional integrity, better is the depolarization. It's something like that. So to show the case example again here, when we compare the normal and an early RP, advanced RP and Stargardt's, as the degeneration progresses, the depolarization signal goes on decreasing. This RP2 patient with relatively advanced, there were hardly any detectable, we could not make out any ellipsoid zone, but still this patient had vision of 6/18. And ERG is quite difficult because of the poor focus, did show reduced central response. But this PSOCT is giving some signals from depolarization signals over here. So indicating that there are some surviving photoreceptor and there is some talking going on between the retinal pigment epithelium and the photoreceptors. That's what the patient is seeing. And these are the patients which can still be eligible for gene therapy, as we heard Professor Holden mentioning. Once everything completely atrophies, if there are no photoreceptors to take up the vectors where we are injecting, there's no point. So like this, the polarization sensitive OCT can be very, very sensitive in both recruiting the patient and also to monitor once we do any gene therapy. So we're talking gene therapy, but where do we, how it is done? Where do we get the vectors from? How to produce that? I think Dr. Arka will brief on the same now from now on. Over to you, Dr. Arka.
Thank you, Poorna. So in the next stage, I will introduce everybody to gene therapy and what we do here in our lab to bring gene therapy to India. So essentially, gene therapy is a method of correcting the pathologic effects by putting back the correct copies of genes. So there are different ways in which you can do this. So typically, we take the normal or healthy copy of the gene and put it back to the cell or the tissue to compensate for the non-functional gene. So this is typically called gene augmentation. You can also fix or correct the genes by either recombination or gene editing or selective reverse mutation techniques. Or you can also express a partially correct copy of the gene through a technique called exon skipping, where you skip over the mutated portion of the gene. The other sort of distinct aspect is changing the regulation of genes. So this is typically applied in case of complex disorders such as cancers or diabetes or AMD or diabetic retinopathy, etc., where the disease pathways which are pathologic in that tissue are going to be regulated by the gene therapy that you do. So we'll discuss all of these as we get along. So in our lab, what we do is we work with this vector called the adeno-associated viral vector. So it is a single-stranded DNA vector or a DNA virus from which we remove the native genes, which is the rep and cap genes, and put our gene of interest in it. Now, this gene of interest is our therapeutic gene. So we package it into the viral vector capsid. And this viral vector capsid is essentially injected into the tissue type that you want to get it to, where it will express the correct protein and give you the therapy. So why do we use AAV? Because this is currently the gold standard for vectors. It is very efficient in delivering the cargo, the therapeutic gene, to both dividing and non-dividing cells. It gives fairly high levels of gene expression. It does not carry any of its own genes, so you're not going to get an immune response to the viral genes. It is typically, amongst the various viral vectors that we have, it is typically much lower in immune response unless you are delivering it at a very, very high dosage. And finally, it is fairly stable if stored correctly for a long period of time and gives an expression for many, many years. So a one-time treatment is possible using AAV. So our areas of focus are various retinal disorders, retinitis pigmentosa, Stargardt, LCA, AMD, and also corneal diseases, but we also do some work on muscular dystrophy, which I'm not going to talk about. So the specific technical focus of our gene therapy program is in three aspects. The first one is transgene engineering. So here we do something which is called dual vectors, or generate mini genes and microgenes of the mammalian genes. So this is because AAV is really small in size, so it has a very limited packaging capacity, and I'll explain a little bit about it in the next slide. The second thing we do is design expression cassettes, so where we can regulate the expression of the transgene for a specific tissue or a specific target cell type, so that you have safety, so it doesn't express aberrantly in other tissue types. And we also do some work on mutant and chimeric capsids of AAV. This is what we do with collaborators. The third, and which is probably a critical development, is to make the clinical grade vector. So this is where we've developed a clinical grade vector facility I will show in a little bit. So in the past, what we've done is we've done these AAV injections in mouse eyes, and this is using a specific serotype called the AAV9 serotype. So a lot of the gene therapies that you're aware of, say Luxturna and all, they use a serotype called AAV2. So AAV comes in all these different flavors. You can use them for targeting different kinds of layers in the eye. So AAV2 is good for RPE, AAV9 is good for various plexiform layers and also the nuclear layers of the photoreceptors. So now I'll explain to you what we do in case of the dual vectors that we work with. So this is something very unique to our lab. And this is to expand the packaging capacity of AAV. So genes such as, say, ABCA4, which Poorna was mentioning for Stargardt disease, is too large to be packaged in AAV, which has a limit of less than 5 kb. So in order to expand that, what we do is we split the gene into 2 parts. And this is the dual vector technology. So you have different ways of splitting it and putting certain signals at the ends of these split parts. And those are the approaches are called overlapping vectors, transplicing vectors, or hybrid vectors, which use both of these techniques. So essentially, if you think about it, so this dog is the piece of DNA which is larger than the AAV genome, and the basket is the capsid, right? So the dog is trying to get into it, can't get into it. So what we do is we take the gene and we make 2 pieces of it. Each of these pieces are packaged separately into AAV vectors, and then we co-infect both of these into the target tissue. So inside the cells, these 2 pieces will come together and express. So does this actually work? So it does actually work. We've already shown this, published it in muscle diseases. And this is just an example of an experiment where we show it in mouse eyes. So if you inject a single half of the gene, which is this first micrograph, you don't see any expression. So the expression here is of an enzyme, beta-galactosidase, which gives you this blue color. And if you have the whole gene there, then you can see the blue color. And these bottom 2 panels show you 2 different kinds of dual vector reconstitution. So you can clearly see that these genes are well expressed in the plexiform and the outer nuclear layers of the retina. So the retina has the capacity to do these recombination events and do these splicing events which are required for dual vector reconstitution. So this expands our ability to use AAV for a variety of different diseases, including Stargardt's. So with that, I will now move to AMD. This is a complex disease, and I will now invite Dr. Thirumalesh to take over AMD.
Yeah, thanks, Arko, and I want you to keep the control of the slides with you. As Arko just mentioned, the genetic AMD is a very complex disease. Single gene disorders are there is a gene defect, and the expression of which causes photoreceptor loss, and the patient loses vision. In AMD, the patients who are getting AMD were normal when they were younger, and because of certain changes in the genetics, there is a genetic component of it where a few genes are abnormal. Say the genes on the slide, like as you can see, are the commonly associated genes. The CFH and ApoE are kind of act at the level of oxidative stress and lipid peroxidation in the cellular metabolism. Once there is a chronic inflammation set up at the cellular level, the CFH gene, which kind of takes part in the complement activation, comes into play. And when there is neovascularization and fibrosis, the VEGF-A and the HTRA1 and ARMS2 genes are, kind of can give us a prelude that these patients are going to end up in advanced AMD. So when the International AMD Genetic Consortium summoned a study, a JIVA study, to look at what genes are involved in AMD, they found a whole lot of them. So they found that there were many genes which were kind of involved in the process of AMD, and these genes were kind of involved in complement activation, collagen synthesis, lipid metabolism, receptor-mediated endocytosis, endodermal cell differentiation, extracellular matrix genes were all associated with patients who are having advanced AMD. So when you look at the genomic aspect of AMD, we feel that it's a very complex disease. Many genes are involved, and many genes at different levels of molecular mechanism are kind of responsible for this disease called as AMD. So let's look at the cellular aspect of it, at what is happening at the RP level in patients who are having AMD. So we all know this particular part, that the photoreceptors tend to shed their outer segments, and the RP is responsible to clear off the outer segments and also generate the activated vitamin A, which is really essential for the visual cycle. So what happens if the RPE kind of gets tired and is not kind of processing the shed outer segments is that the engulfed part of the photoreceptor outer segment is processed in the lysosome. So whenever there is a failure in the lysosome activation and it's not doing its work properly, what it does is it kind of sets in an inflammation within the cellular level, and this is called as which initiates a process called as an inflammasome activation. And the continued process of processing of the outer segments kind of causes deposition of drusen, and the continuation of which also associated inflammation which is there within the cell will cause damage at the level of the RPE, at the level of the choroid capillaries, and kind of takes the disease to the next level where they end up with choroidal neovascular membranes and fibrosis at all. So to understand the disease, one has to put together what is happening molecularly, because majority of us as clinicians, whenever we say there is an inflammation, we want to look for cells and flare in the milieu. But the molecular signature of inflammation is already there in the disease. So when we studied a cohort of patients where we took aqueous samples from patients who were undergoing injections in CNVM, because that was easily available, what we saw that there were, along with the classical angiogenic factors which were definitely there was presence of a lot of cytokines, chemokines, and soluble adhesion molecules. So our question was, who is producing these soluble factors? A partial guess is that the RPE is having a cellular level of inflammation, but does it causes so much of inflammation, or is there a homing of selective homing of activated neutrophils? So that is what we wanted to study in this particular cohort. The second thing that we wanted to see was that does it happen in early AMD as well? So what we did was we took aqueous from patients who were undergoing cataract surgery and who did not have any evidence of AMD. And the second cohort that we studied was patients who were having intermediate level of AMD. What we saw was that in the upper part of the graph, you can see, although the number of leukocytes which we were able to see was relatively similar, but in the cohort of AMD, we did see that there was a presence of leukocytes which were kind of activated. So why does this activation takes place, and can this be replicated in an animal model is what we were thinking about. So next slide, please. So then for the animal model, we look forward to our collaborators in Pittsburgh, Dr. Debashish Sinha, along with Arkasubhra Ghosh, to design this particular experiment. So what we did in this particular experiment was that we took the immune cells and we tagged them so that it would be easy for us to image when we do the scan. And we injected the cells intravenously into the NOD-SCID mice, and then we imaged using a ribbon scan. So what happened in the ribbon scan was that the white specks which we can see are the neutrophils, and play the video. Yeah. So you can see that clearly the neutrophils which were activated were predominantly it violated both the blood aqueous and the blood retinal barrier, and we were able to see them right from the anterior hyaloid just behind the lens in the AC. This is in the anterior hyaloid. You can, when you go forward, you can see that the cells are also there intraretinally. The cells were also there in the subretinal space, and the cells were also found in the hyaloid just behind the lens. And if you look closely, even at the ciliary body level and in the pars plana zone, you do see that these cells are present. So this kind of gives us an insight that the eye, what we thought was immunoprivileged, is not immunoprivileged once there is activation of these neutrophils. So this is a final image where we have mapped all of them, and you can see that from the anterior segment to the posterior segment, you can clearly see that the inflammatory cells can reach various locations of the eye, starting from the ciliary body up to the retina and the optic nerve. Next slide, please. So based on this, we have established that AMD has a genetic angle of it, which is pretty complex. AMD also has an inflammatory angle to it, which is pretty complex. And we also have a key indicator of AMD progression is that there is a lysosomal dysfunction. So based on this, we can kind of come to this, designing of therapeutic strategies for AMD, and choosing the right target is really essential. So based on the previous slides, we can conclude that one should work on modulation of the immune status. One should also work on regulation of the secreted factors and immune cells, because once they come in, they tend to generate a lot of factors which are soluble. One should also look at rejuvenation of the RPE, because the RPE is one cell, which is getting tired as we age, and which then affects the autophagy and the lysosomal pathways. So our collaborators and us are kind of working on this particular mechanism, where we are trying to enhance the lysosomal pathways so that it can be applied to treatment as and when the trial comes forward. So why are we doing all this research? Is because we know that there is no treatment in dry AMD. So all we have is a bunch of vitamins and supplements that we tend to give to the patients, and we are silent observers as the patient progresses. And also newer treatments are required apart from anti-VEGF in patients of neovascular AMD, because after a certain point of time, the neovascular AMD patients tend to become refractory to therapy. We also require long-term solutions which are required, so because of the chronic nature of the disease, because long-term monthly or bi-monthly injections might not be feasible in economic zones like us. And one should also look at bioavailability in the eye over longer duration for proper disease control. This is because there are a few groups who are working on generating an anti-VEGF molecule by doing genetic engineering within the eye. That kind of is a very exciting kind of an aspect to AMD. But in this enthusiasm of bringing in gene therapy and cell therapy to reality, we should always consider that we should not be causing significant immune reactions in an already immune heightened environment, which is AMD, where there is already a baseline in immune activation is already happening. So with this, I think I'll hand back to Arko to go to the next part of it, where he'll be illustrating much more on it. Thanks, Arko.
Thanks, Thiru. So we discussed how we are approaching inherited disease and complex diseases separately, and how we go about target selection in this. So how about the cornea? So cornea is another very potent target because it's easily accessible, it can be easily monitored. So this is just an example of corneal gene therapy that I did with Professor Rajiv Mohan many years ago, which shows how the different serotypes of AAV work in the mouse cornea. So with that, I will invite the Pooja. So she will discuss cornea and how we go about selecting genes for corneal diseases. Pooja, you have control.
Thank you, Dr. Arka. So keratoconus is one disease where So keratoconus is one disease where it leads to gradual progressive diminution of vision, where there is steepening of cornea, and it is also known as a corneal thinning disorder. Now, initially it was known to be a non-inflammatory disease, but various groups, including ours, have proven that now it's an inflammatory disorder. The incidence is pretty much higher, where it affects 1 in 430 to 2,000 across the globe. But most interesting part is that incidence in South and West Asian populations, including India, it's 4.4 times higher than the other countries. We do have various treatment options like corneal collagen cross-linking for the early stage of the disease, and if it's advanced, we can do intracorneal ring segments and corneal transplants. But however, the interesting part here is that even after corneal collagen cross-linking, we have seen that there can be progression of the disease. And another interesting part is that even if, the second part would be to do a transplant. Even after transplant, there have been various case reports which talk about progression of the disease. Now, after transplant, where you have implanted a healthy tissue, a healthy donor cornea, why will it lead to progression? It is because the disease is migrating from the diseased host to the donor cornea, which might be because of the repopulation of the affected keratocytes, which is leading to the aging of or the affection of the graft tissue, and which might lead to failure of the cross-linking. Now, looking at all this, various studies, as mentioned, like, inflammatory in nature, there are various components, like how AMD has various components, even keratoconus has various components. It can be inflammatory cytokines, There can be involvement of matrix remodeling factors like lysyl oxidase and collagens, which we would come in the next few slides, or it can be related to genetic mutations as well, and which has been studied. So this throws a light on that, why gene therapy is important when it comes to keratoconus disease. Our group has been trying to do various studies where trying to study the keratoconic families where there is a proband, and the families also have some subclinical disease which has not manifested. And what we have found is that there are interestingly there are new set of genes which are seen just in these diseased families where they don't have, the parents don't have a manifest disease, but the children have it, and which is not seen in the controls, in the families and in the probands which do not have disease. So it throws a highlight that, again, the future in keratoconus management is going to be gene therapy. We have done a lot of work where we are trying to, create a model of what happens, and it's like an immune corneal remodel, where you can see that the various markers like NF-KB and MAP kinase are involved in upregulation of these markers, which are responsible for generation of the inflammatory markers, because keratoconus is inflammatory in nature. But the more interesting part is, more than this inflammatory markers, if you can see the base there is lysyl oxidase and collagens. So if there is reduced expression of LOX or collagen, then none of this is going to work, because you need to take care of the root cause, which is the LOX and collagen, before trying to do anything else. And that's why the LOX and collagen is important. Now, why LOX? Lysyl oxidase. Lysyl oxidase is nothing, but it is an it's a cross-linking enzyme where it holds the collagen fibers together in your corneal stroma. However, if there is reduced expression of lysyl oxidase, your collagen fibers in the stroma are going to start slipping away, which is going to lead to reduced stiffness of the cornea, which will lead to keratoconus or an ectasia disease. And in this cornea, if you do a cross-linking or a transplant, it might have a failure. So the question here is that maybe when we increase the expression of LOX, the collagen is going to be cross-linked. It is going to maintain the stiffness of the cornea and the extracellular matrix remodeling. How can we say this? Because we have seen that in the graft tissues of patients, of keratoconic patients undergoing transplant, and from the healthy donor corneas, we have seen that in the keratoconic eyes, there is reduced expression of LOX. You cannot see, if you can see the brown color here in the healthy donors, that means the LOX is present, which is absent in the keratoconic eye, in the stroma, and which was further even studied in the corneal epithelial cells, that the lysyl oxidase was lower in quantity compared to that of controls, and that trend was maintained with increasing grades of keratoconus. And also the same was studied in the tears of the patient, suggesting that yes, okay, sir, can you just move the slides, please, because I don't think it's happening.
Yeah. Go ahead.
And the same was studied in the tears as well. So next we wanted to study that whether, we know that there is a cone present in the cornea and the surrounding area looks healthy on topography. So we wanted to see whether the cone area has reduced expression of LOX compared to that of the periphery. And yes, our hypothesis held true here as well, that the diseased area had reduced expression of LOX compared to that of periphery. Now we have been talking about, and we have found that LOX is the culprit. So when we do a cross-linking, whether the same, that means that, if you do a cross-linking in an eye with a good amount of LOX, the outcome should be optimal. And if, next slide, please.
Yeah. It's okay. Go ahead.
Okay, so that means that, if there is a good amount of LOX present in your corneal tissue, that means the cross-linking outcomes might be optimal. And if the collagen and the LOX levels are lower in the quantity in your cornea, then the cross-linking outcomes are going to be suboptimal, and which has been proven by our group as well. That higher the quantity of LOX, better your cross-linking outcomes, and lower the quantity of LOX or collagen in your cornea, suboptimal the outcome. So how do we take this forward? We did some studies in the stromal cells of healthy patients and the keratoconic patients as well, where we did a gel contraction assay. And when we did that, we saw that, in the healthy controlled eyes, the gel was contracting. And in the keratoconic patients, the gel was not contracting. That means that there is decreased ability of the cross-linking in the collagen bed of the keratoconic eyes. So now there is reduced stiffness in the keratoconic patients compared to that of healthy controls. Now, when we inject the stromal cells of the keratoconic eyes and the healthy cornea with LOX vectors, what we observed was that in the cell controls that there was and injecting the LOX in the keratoconic stromal cells, there was contraction of the gel, which suggests that there is increase in the strength of the collagen bed and the increase in the corneal stiffness. So how do we proceed further? The next step would be to do this LOX vector therapy in the animal studies, and the way forward would be to do clinical trials on the same and in patients. Over to you, Dr. Arkasubhra.
Thank you, Pooja. So essentially, we developed some vectors, both lentiviral and adeno-associated viral vectors for expression of LOX. And when we tested it in animals, it was a simple procedure where you debride the epithelium and just give it as an eye drop. And that leads to significant expression of the protein. But interestingly, it also leads to, so this is the mRNA expression from the corneas, from these animals with treatment of AAV expressing LOX or a control AAV virus that express GFP. So you can see that even the collagen expressions have gone up, and this is something that we have shown in the past in vitro, that both TGF-beta as well as lysyl oxidase overexpression can lead to extracellular matrix proteins being upregulated. Something interesting that we also see is that the basal level of MMP9 also reduces, and more functional studies are underway right now. So apart from what we are trying to do in keratoconus in the past with Professor Rajamohan, we've done studies on understanding how we can reduce post-surgical haze in case of corneal refractive surgery. So this is a study with AAV expressing SMAD7, which is an inhibitor of the TGF-beta signaling pathway. And what you can see in rabbit eyes is that the expression of AAV SMAD7 significantly reduces the post-surgical haze. So therefore, the application of AAV delivered genes can be taken to trials from all of these animal studies that are ongoing. So, but in order to go to trials, you have to make the clinical grade vector. So what we've developed in Narayan Nethralaya is a clean room facility, which is now equipped to do, in a small scale, generate the clinical grade vectors, which can be used in a clinical trial. So you can see the workers here, they're all fully gowned. And there's a series of procedures for going in and coming out of the facility. So with this, another important step that has happened in India is that in end of 2019, the national guidelines for gene therapy clinical trials was published by the Indian Council of Medical Research and the Central Drug Standards Control Organization. So this is an important step that enables us to start gene therapy clinical trials. So to summarize, our knowledge of genetics and the feasibility of doing genetic testing has opened the door for gene therapy. The gene therapy trials in India have to be done with regulatory approval from GTAEC and SPIDESCO. So in the audience, whoever is listening to, please remember this. The choice of a gene for the therapy is critical, and as our studies are showing, and studies that Dr. Will Pandian also talked about, it's very important to identify the node at which you are going to intervene, and which gene would that be. Now, making clinical grade vectors or the drug is the key bottleneck, which we are now solving here at Narayan Nethralaya. And as Dr. Sharma has pointed out, clinicians, scientists, genetic counselors, everybody needs to work together to establish the roadmaps for personalized medicine and make these therapies available in India. So with that, I'll thank everyone and thank my team, both the Grow Lab as well as the entire family of Narayan Netralia. Thank you.